The Protein Data Bank includes 3D structures of all biological macromolecules, including proteins, nucleic acids, carbohydrates, lipids, and their various complexes. Lipids are important biological molecules. You see them in membranes as a storehouse for energy, as hormones, and as key signaling molecules. They are made up of building blocks like fatty acids, alcohols, steroids, that are covalently linked together with each other to form glycerol esters, fats, waxes, phospholipids, etc. But these molecules do not form long polymers like proteins, nucleic acids, or carbohydrates. Instead, lipid molecules interact with each other to form membranes, rafts, micelles, etc. to exclude water molecules and perform many of their functions. Unlike proteins, nucleic acids, and carbohydrates, their structure is often not ordered. Also, the high proportion of carbons and hydrogens in these molecules do not provide enough contrast to be viewed in electron microscopy images. While the binding of specific small molecule lipids to a binding pocket on a protein may be ordered and visualized like any other small molecule ligand, the case of transmembrane or membrane-bound proteins is different. Since they are intimately linked with membranes for their structure and function, they require the presence of lipids in order to be purified and/or studied. To keep the protein stable and soluble in the experimental sample, crystal structures of membrane proteins include detergent molecules like beta-octyl glucoside. These molecules have both a polar and a non-polar portion. While the non-polar portions mimic lipids and help stabilize the protein, the polar parts make the complex soluble in water or buffer. So structures of membrane proteins often include detergents and small hydrophobic molecules bound non-specifically to the region where the membrane would bind. In larger structures of membrane-bound complexes, the sample may include micelles or parts of a liposome to mimic the membrane. In some cases, these membrane mimics are part of the structure coordinates and can be visualized in atomic detail, while in other cases, their presence is implied and can be seen when viewing experimental data like EM maps. Keep these ideas in your mind as you explore the various membrane proteins and other lipid-binding proteins in the PDB.